안녕하세요. 저는 현재 시카고 대학교에서 유전학 박사과정 2년차 밟고 있는 김현경이라고 합니다. 박사과정을 시작하기 전에 제가 이 매사추세츠 일반 병원에서 진행했던 당뇨병 연구 논문이 지난 3월에 게재가 되어서 오늘 그 연구에 대해서 발표하려고 합니다. 발표는 영어로 진행하도록 하겠습니다. Hi everyone, I'm Hyun-Kyung Kim. I used to be a bioinformatics analyst working at the Mass General Hospital and currently a PhD student in genetics at the University of Chicago. Today, I'm going to talk about my work at MGH and Broad Institute, titled High Throughput Genetic Clustering of Type 2 Diabetes Loci Reveals Heterogeneous Mechanistic Pathways of Metabolic Disease. This project has been published in the March issue of Diabetologia, so you can find the article using the QR code below on the bottom right of the slide. I will go through the background, methods, results, and then summarize the key points. To begin with, diabetes is one of the most common complex diseases, afflicting approximately 460 million adults worldwide. Type 2 diabetes accounts for about 95% of diabetes and influenced by multiple risk factors, including genetics. Despite the recognized heterogeneity in patient phenotypes and responses to treatment, Type 2 diabetes management strategies remain largely impersonalized. In an attempt to deconstruct the heterogeneity of Type 2 diabetes, recent studies have performed cluster analysis of individuals using serum biomarkers and clinical data like age, BMI, and hemoglobin A1C to identify Type 2 diabetes subgroups. However, these variables change with disease progression and treatment. Additionally, it is not clear whether these clinical biomarkers are causal, consequential, or coincidental in the disease process. Also, selection of biomarkers could introduce subjectivity or bias. On the other hand, genetic biomarkers do not change throughout lifetime and unaffected by treatment or disease course, which seems more suitable for finding causal mechanisms. Since genetic biomarkers are rooted in mechanisms, it can guide inclusion of biomarkers, which is more agnostic approach. Over the past decade, genome-wide association studies and other large-scale genomic studies have identified over 300 loci associated with type 2 diabetes. But for most of these loci, the variants and the mechanism that cause type 2 diabetes remain unknown, limiting opportunities for clinical translation. This is because it is often unclear which variants are functional and which genes are regulated. We can bypass those challenging steps with multi-trait cluster analysis. However, standard clustering approach in the field, like unsupervised hierarchical clustering, is a hard clustering method, whereas it assumes each locus belongs to just one cluster or pathway, and this is not consistent with complex disease biology. So we want to use a more flexible approach that would allow a variant to impact more than one biological pathway to improve cluster interpretability. So the prior work in our group in 2018 used a soft clustering approach called Bayesian non-negative matrix factorization, in short, BNMF. This study used 94 variants that were previously published, restricted to those with a nominal level significance in European populations, and 47 relevant traits with publicly available geoisomer statistics. Those traits include glycemic traits, anthropometrics, and additional labs to better characterize and understand the clusters. For the 94 variants and 47 traits, a variant by trait association matrix was generated as an input for the BNMF clustering. BNMF clustering factorizes the input matrix into two matrices, which are K clusters of variants and K clusters of traits, with an optimal number of K. K is the number of clusters. K is determined using a Bayesian framework such that we see how many clusters are the best fit given the data. This method allows variants and traits belong to more than just one clusters, which reflects the biological mechanisms where genetic variants and traits involve in multiple pathways. In using that framework, 5 clusters were identified in more than 80% of iterations. These are the 5 clusters identified, each of them with distinct mechanisms and loci. First of all, you can see that the betting insulin trend are separating the 5 clusters into 2 groups, which are the first 2 clusters and the rest 3 clusters. The first 2 clusters are more related to insulin deficiency, while the rest 3 are more related to insulin resistance. Key traits and genetic loci are most strongly weighted loci and traits, and based on these key traits and loci, the clusters were named as beta cell, proinsulin, obesity, lipodystrophy, and liver lipid. Here, I won't go into too much details for now, since we have the updated results. So we came up with a question that, can we find more genetic clusters of type 2 diabetes by extending variants and traits using an automated pipeline? And this is why we performed a clustering analysis with expanded loci and traits using an automated pre-processing pipeline for extracting variants and filtering and traits from GWAS datasets. So I will introduce our automated pipeline along with our latest results. This automated pipeline takes multiple disease GWAS of outcome of interest. Here is type 2 diabetes, and it pre-processes the variants by replacing multi-allelic, ambiguous variants, and performs LD pruning. For traits, the pipeline filters the traits by the minimum p-value cutoff across a selected set of variants in order to include only the relevant traits, and then filters correlated traits so that we get independent set of variants and traits. After this process, the pipeline generates a variant by trait association matrix as an input for the BNMF clustering analysis. For the latest results, these are the final traits that were included in the clustering of type 2 diabetes variants, including glycemic, anthropometric traits, and vital signs and additional labs to better understand the clusters. After applying the pipeline, there were 323 variants and 64 traits used in the clustering analysis. For comparison, the previous publication had 94 variants and 47 traits. Now let's jump into our latest result. The convergence was the best for k equals 10 in 38% of iterations. So we used k equals 10 for the downstream analysis. These are the 10 clusters, and 5 of them are relatively new compared to the 5 clusters in our prior work. Beta cell cluster divided into 2 separate clusters, and we have additional lipoprotein A, ALP negative, SHBG, and hyperinsulin secretion clusters. Lipoprotein A is driven by high lipoprotein A, ALP negative by reduced ALP level, SHBG by reduced X-hormone binding globulin, and hyperinsulin secretion cluster by high disposition index and corrected insulin response. Here we have categorized the clusters by association with fasting insulin. 4 clusters in the red box, the 2 beta cells and proinsulin and lipoprotein A clusters were associated with reduced fasting insulin trend. And another 5, including obesity, lipodystrophy, liver lipid, ALP negative, and hyperinsulin secretion clusters, are associated with increasing fasting insulin. And SHBG cluster is unclear since it has no significant association with fasting insulin. So we'll have a quick look at the clusters. Here, the green bars are showing the weights for the key loci, and red bars represent the increased trait and blue for the reduced trait in each cluster. For those of you who don't have much background in diabetes biology, beta cell produces insulin, which controls the level of glucose in the blood. And proinsulin is a precursor for insulin. The 2 beta cell cluster have key genetic loci that includes many well-known beta cell related loci from the beta cell cluster in our prior work. In the proinsulin cluster, ARAB1 and STAR-D10 region has previously been connected to impaired beta cell function in mouse models. Obesity cluster includes well-known obesity-related loci like FTO and MC4R. And also key traits include increased BMI, body fat percent, waist and hip circumference. And so the suspected mechanism for this cluster is obesity-mediated insulin resistance. On the other hand, HDL level and insulin sensitivity index is reduced in the lipodystrophy cluster, and the loci in the cluster are known to be involved in lipodystrophy or fat distribution-mediated form of insulin resistance. The liver lipid cluster includes GCKR and PNPLA3. which were previously associated with non-alcoholic fatty liver disease, NAFLD. Now let's look into more details with more focus on the newer clusters identified in this work. Again, insulin deficiency and insulin resistance categorization is using the association with fasting insulin. The two beta cell clusters and proinsulin cluster share the reduced HOMA beta, which means that beta cell is not working well in these clusters. Beta cell 1 and beta cell 2 have many loci that overlap, and some of them are MODY genes, which are monogenic diabetes, like GCK. Lipoprotein A is associated with increased LPA, and SLC22A3 is known to be associated with plasma lipoprotein A levels. Among the 5 clusters related to mechanisms of insulin resistance, 3 were captured in our prior analysis, but gained additional loci in this expanded analysis. Obesity cluster, again, includes some well-known obesity loci like FDO and MC4R. Lipodystrophy has ILRS1 and KLF14 and PPAR gamma as the top loci, and decreased HDL insulin sensitivity index and increased triglycerides, suggestive of fat distribution-mediated insulin resistance in the previous studies. Liver lipid cluster includes multiple loci previously connected to hepatic lipid or glycogen metabolism, including GCKR, HNF1 alpha, and PNPLA3. And functional work has implicated these loci were associated with isolation of lipid in the liver, resulting in decreased serum triglyceride levels. And we can see that triglyceride level is reduced in this cluster. Triglycerides are the lipid that give your body energy. And ALP negative is associated with reduced alkaline phosphatase and driven by ABO locus. ABO locus and blood type have previously been connected to type 2 diabetes risk, but the causal mechanisms are not fully understood. And hyperinsulin secretion cluster is associated with increased disposition index and corrected insulin response. And more than 87% of the loci in this cluster were not the part of the previous analysis, which is probably why we were able to identify this cluster in this expanded set of loci. SHBG is associated with reduced sex hormone binding globulin and driven by SHBG locus, single locus. And our results point to a genetic pathway whereby alteration of the SHBG locus leads to reduced sex hormone binding globulin levels and type 2 diabetes risk, which is consistent with previous epidemiological and genetic studies indicating that low circulating levels of sex hormone binding globulin were causally related to increased risk of type 2 diabetes in both sexes. Since we're not completely sure of the mechanisms of the new clusters, we performed some downstream analysis. The first downstream analysis was performed is glycemic categorization of clusters using GWAS summary statistics. And we call the effect sizes of this analysis as cluster-specific GWAS partition polygenic scores, in short, GWAS PPS. The idea here is to combine the effect of each of the top variants in a cluster to get the association effect size for the cluster. This is where the insulin deficiency, insulin resistance categorizations you've seen in the previous slide came from. The two beta cells, proinsulin and lipoprotein A clusters, have associations with reduced fasting insulin levels, which is colored as light green here, whereas the other 5 clusters have association with increased fasting insulin level, except the SHBG cluster, which has no significant association. Beta cell 1, GWAS PPS, had a more marked association with a reduced disposition index compared to beta cell 2, which is colored as blue, while beta cell 2 had a more marked association with increased proinsulin level, which is colored as green here. And this indicates that beta cell 2 relates to more specifically to beta cell stress. Also, we can see that fasting insulin association separates the two beta cell and proinsulin cluster. And that potentially indicates a mechanism of lack of proinsulin substrate for insulin synthesis and secretion for the proinsulin cluster. And hyperinsulin secretion cluster is the only one that is associated with increased disposition index, the blue. And disposition index is the product of insulin sensitivity times the amount of insulin secreted in response to blood glucose levels. So we've seen the glycemic characterization of the cluster so far, and now we'll move on to the cluster associations with clinical outcomes. So we were interested to see if the clusters are differentially associated with disease outcomes. First is coronary artery disease, and second is chronic kidney disease. The analysis in plot A also involves using the GWAS summer statistics, similar to the glycemic associations in the previous slide. On the other hand, plot B was generated using the individual level data in Mass General Brigham Biobank. There were a total of 25,000 individuals with genetic data from European ancestry, and there were about 2,000 type 2 diabetes cases. For each individual, cluster-specific polygenic scores were generated by multiplying a variant's genotype dosage by its cluster weight, with only the top weighted variants included. Also, the association with lipodystrophy cluster and risk of CAD was confirmed in both analyses. CAD is in the red dot here. But the association between liver lipid cluster and the risk of CKD, which is the black dot here, was directionally consistent, but not significant in the individual level analysis. In order to define cluster-specific mechanisms of each cluster, we performed epigenomic enrichment analysis using tissue-level and single-cell level data. We expected that each pathway to capture a different disease mechanism and thus localize largely to specific and distinct tissues. So we assessed whether variants in each cluster altered enhancers or promoters in specific cell types. Several clusters displayed tissue-specific epigenomic enrichment as shown in the heat map, plot A on the right. For example, the 2 beta cell and proinsulin clusters were enriched in pancreatic islet tissue, lipodystrophy cluster in adipose tissue, and liver lipid and ALP negative cluster in the liver tissue. Moreover, the loci in the 2 beta cell cluster shown on the left were differentially enriched in functional and stressed pancreatic beta cell states in the single cell level data shown on plot B. Here, insulin high and insulin low indicates high or low insulin gene promoter accessibility. So the insulin high islet cells had enriched promoter accessibility for genes involved in insulin secretion, whereas the insulin low cells had enrichment for the stress-induced signaling response genes. And so this analysis suggests that we are grouping together loci in clusters that act in particular tissues or cell types. Those were the results, and now we'll go to the conclusion. For strength of this work, we've expanded clustering with more variants and traits with automated pre-processing pipeline, and now we have 10 clusters, including previously identified clusters, and the newly identified cluster were driven by traits and loci that were not available in the prior analysis. Also, a couple of those clusters were shared with clusters of CAD and CKD in separate analysis. For limitations, this study includes clustering of only available phenotypes from GWAS, and genetic variants used in the cluster analysis were restricted to those associated with type 2 diabetes in populations of European ancestry. Also, we're uncertain whether all weights or a thresholded approach is ideal for assignment of elements to a cluster. Now I will summarize the key points of our work. First, clustering of genetic variants associated with type 2 diabetes has identified 10 robust clusters with distinct trait associations, which likely represent mechanistic pathways causing type 2 diabetes. These clusters group together loci in clusters that act in particular tissues, and clusters were associated with distinct clinical outcomes. High throughput pipeline that we used can be applied to other complex disease beyond type 2 diabetes. For future work, we could expand to non-European populations, which is part of the ongoing work. And we'll also assess the impact of clusters throughout the disease course using some time series data. So that is all I have. And with that, I'm happy to take any questions.